In this session, we will understand about the discovery of DNA as the molecule of heredity or as the genetic material. Right now, the field of genetics has a rich past, and the earliest of the discoveries, the most significant of the earliest of the discoveries, was made by Mendel. Now, you people must have studied his experiments in your previous classes where he worked with bees and was able to actually figure out that the inheritance, that whatever we inherit from our parents is not a blend of their traits, and the information is carried by certain particles, as he called them, from one generation to the next. So Mendel was the first one to actually theorize and postulate that there were certain particles present inside the cells which were basically contributed by both the parents, by the father and the mother, and these particles then were inherited by the offspring, and then these particles were responsible for the characteristics or the traits. That was Mendel's contribution. And what did he call these particles? He called them as factors. He named them as factors. And today what we know of factors is that factors are just genes. He was talking about genes, but he had no idea at the molecular level what these factors were. He could only deduce by looking at the phenotype, or by looking at the characteristics expressed by the pea plants, that there was something responsible for the expression of those traits. Similarly, there were many other individuals who had contributed greatly to the field of genetics. For example, this individual called Miescher. Now, Friedrich Miescher was basically a biochemist, if you would like to say it that way. Now, he was a biochemist who was for the first time was able to chemically isolate the nucleic acid DNA, and he called it nuclein. He was for the first time able to isolate it, that this is the substance which carries the genetic information, but nobody still knew what was the chemical nature of that particular substance, which he called nuclein. He called it nuclein because he isolated it from the nucleus, but he had no idea whether it was a protein, whether it was DNA, whether it was RNA, whether it was a carbohydrate, or whether it was a lipid. He had no idea about that. Similarly, another contribution was made by Sutton and Boveri, who proposed the chromosomal theory of inheritance, according to which the characteristics are transferred from one generation to the next by chromosomes. So I hope you remember what chromosomes are. These are highly folded, condensed structures, which basically is nothing more than chromatin or coiled up DNA. So they also proposed the theory of chromosomal inheritance, because by that time there was a microscope, and these individuals, they observed the cell division events, and they could actually see the chromosomes moving from one cell to the other and into the daughter cells, and they were the one who postulated the chromosomal theory of inheritance. But in spite of all of these advancements, all of these discoveries, the molecular basis of genetic material were unknown, which means what particular molecule was actually carrying this genetic information. And there was this major view that proteins were the major contenders. Proteins were thought of as the major contenders of carriers of heredity. There were many other indirect physical evidences that pointed towards DNA as the molecule of heredity, but then there was what was lacking was the experimental proof. And this is where the three major experiments that we are about to discuss came into play, which led to the discovery of DNA, which concluded DNA is the genetic material. And now let us look at all those three experiments one by one, and let us just start and let's proceed with our first one. So this is our first experiment that we are going to study, and this experiment was conducted by Frederick Griffith. And here is this gentleman, Mr. Frederick Griffith. And Frederick Griffith was a British bacteriologist who was working on bacterial pneumonia. Now, pneumonia is a disease of the lungs. Right now, he was working on bacterial pneumonia caused by a bacteria called Streptococcus pneumoniae. And he was primarily working on wide aspects of Streptococcus pneumonia, or this disease called pneumonia. He was studying the epidemiology, which is He was studying how this particular disease spreads around, how it moves around, how does it survive, what are the different vectors if there are any, etc., etc. He was also working on the pathogenesis, which is how this disease develops in a particular organism. So he was pretty busy with this particular bacteria. And he was also involved in serotyping of these bacteria, which means he knew greatly about the different strains of these bacteria. He was working with large number of strains. Now, how did he identify the different strains? Was he made use of different antibodies that each different type of Streptococcus pneumonia produces in the body. So he was able to basically figure out that there are different types of Streptococcus pneumonia bacteria. Now, he chose two strains of Streptococcus pneumoniae for his experiment. So the first strain was called the rough A virulent strain, and the second strain that he chose for his experiment was the smooth virulent strain. Now these two strains differed from each other in a certain very important aspect, and let us just have a look at that. Let me just clear up the board just a bit. And so here what we see is we have a petri plate. It's a glass dish. It's a plate made out of glass, inside of which we grow microorganisms. So in this particular plate, this is called a blood agar plate, which means it has a nutrient medium inside agar. Now agar is a solidifying substance. You can imagine it's something like a jelly inside which we have a variety of nutrients, and one of those nutrients is blood, and that's why it's called the blood agar plate. Now this particular material, blood agar, supports the growth of Streptococcus pneumoniae, various strains. Now if this rough avirulent strain is cultivated or cultured on this blood agar plate, the colonies, when I say colony, I mean millions of these bacteria, the Streptococcus pneumoniae, they don't grow isolated, they grow as a mass and they form something called as a colony. And here, if I magnify it just a bit, let's just magnify it 1, 2, 3, 4 times, and you can see, actually now you can see that, I hope you can see this colony. So these are different colonies, you can see these different colonies. Now let's also go back. Yeah. So these colonies, when this rough avirulent strain was cultivated, let's just demagnify it just a little bit so that I have the entire screen for myself. Let's bring it a little down. Okay. Here we are now. So when he cultivated the rough avirulent on this blood agar plate, he observed rough colonies. When I say rough colony, I mean to say that the edges of this colony were irregular. They were not smooth. So this is one of the colonies. Inside a colony, you will have like millions and millions of these Streptococcus pneumonia bacteria. Now, when this smooth virulent strain of Streptococcus pneumonia is cultivated on this blood agar plate, we will get smooth colonies, and a smooth colony has smooth edges. It does not have a wavy or a rough edge. So these were smooth colonies. Now, why the smooth colony was smooth? It was smooth because the Streptococcus bacteria of the smooth strain, and let me draw a coccus bacteria first. So coccus is a spherical bacteria. Okay, so this is a bacteria. Now don't confuse the two. This is a colony. This is a group of like millions of these bacteria, and here I've just drawn one cocci or a spherical bacteria. Coccus is a sphere. Now this in the Streptococcus pneumonia of the smooth colony had around them a capsule. They had a covering around them, and this covering is called a capsule. And this capsule is made up of a polysaccharide. And this polysaccharide, the importance of this polysaccharide around this smooth strain of Streptococcus pneumonia is that it confers immunity or protection. It gives this bacteria protection against the cells of the immune system of the host. Now, for example, if this bacteria was to enter the host, let us say a human being, once this bacteria is inside the human body, the immune system of the human body will come into action. And there are these cells called the lymphocytes, the cells of the immune system. Once an example of a lymphocyte being a neutrophil. Now, neutrophils have this ability, are phagocytic in nature, which means that they can engulf a foreign particle, digest it, and that is how they protect the individual from pathogens. So these neutrophils, when they identify this particular pathogen, they are unable to engulf it. Why? Because this polysaccharide layer protects. Now let us assume that this is a macrophage that I have made. It is trying to engulf this bacteria, the smooth strain of Streptococcus pneumoniae. It is unable to engulf it and digest it because it is protected by this polysaccharide capsule. And this polysaccharide capsule makes the appearance of this colony as smooth. On the other hand, if you look at the Streptococcus pneumonia bacteria from one of these rough colonies, you will only find the bacteria and no polysaccharide capsule. Now, because they do not have any polysaccharide capsule, when these bacteria, when they enter the body of a human being or any other mammal which has an immune system, they are very rapidly destroyed by the neutrophils. They are engulfed inside and they are destroyed. Why are they destroyed? Why are they engulfed? Because they do not have a protective polysaccharide layer. So this is the basic difference between the rough avirulent and the smooth virulent strain of Streptococcus pneumoniae. Now let me also emphasize on the terms avirulent. Avirulent is non-disease causing strain, whereas the term virulent means disease causing. And here in this micrograph you can actually see, here in this particular micrograph you can see the capsule, this white blank area around these cocci is basically the capsule. Now let us proceed further. Now let us have a look at what experiment did Frederick Griffith perform with these two strains of the bacteria. Now let me just clear up the slide and let's proceed with this once again. So what Griffith did was he chose mice for his experiment. He chose mice and he chose the smooth strain, let's call them SS, and he chose the rough strain of Streptococcus pneumoniae for his experiment. So these were the materials that he used for his experiments. Now the first of all, what did he do? In the first step, he took, so let's look at this. This is the first step of the experiment. He took living smooth cells, he cultured them, he grew them on a petri plate, and then he isolated all of these living smooth Streptococcus cells which were like virulent. I hope you remember that the smooth strain is virulent, it can cause disease. He took them, isolated them, put them inside a syringe and injected these smooth strain of Streptococcus pneumoniae inside the mouse. Now, as a result, the mouse contracted the disease pneumonia and it died. The mouse died. Common sense, right? And once the mouse had died, he took a tissue sample, a bit of tissue sample, and he put that tissue sample onto a petri plate, and he could see that the smooth colonies of Streptococcus pneumoniae could be isolated, which meant that these bacteria went in, they stayed in, caused pneumonia, and then they could isolate these bacteria once again from the dead body. Right? So this was the first step of the experiment. In the second, now what he went a little further, and what he did, he took the living rough cell. Now the rough cells cannot cause any particular disease. So here's this petri plate with the rough cells on it. They have not drawn it, so let us just draw a couple of rough cells. Oh, this is really messy. So let us assume that these were the rough colonies. He scraped them out, put them into a certain solution, put them into the syringe, and then injected this particular solution into the mouse. Now, as these rough cells are avirulent, they do not have a polysaccharide layer. The immune system of the mouse identified those particular rough bacteria, the Streptococcal pneumoniae bacteria, and destroyed them, and the mouse remained healthy. And from this healthy body, after a certain period of time, a certain tissue sample was taken, and not very long after the injection or infection, a certain tissue was taken and a culture on the agar plate was made, and he was able to isolate the rough colonies, right? But the mouse had survived because these were non-virulent strains. Now he went a step further. In this third step, now he went for the third step of the experiment, and let me choose a different colored marker, and yep. In the third stage of the experiment, what he did, he grew these smooth cells, the virulent disease-causing cells. He took them in a test tube and he heat killed them. He killed these bacteria by applying heat. Now, dead bacteria cannot cause any disease. So what he still did, he took those heat-killed smooth cells and put them into the syringe, injected it into a mouse, and the mouse remained healthy. And now also he took a tissue from the tissue sample or some kind of a blood fluid from the body of the mouse. It could be the blood, it could be something else. And he poured it onto the culture plate once again, and there were no colonies isolated. Why couldn't he isolate any colonies? Because all the cells were dead. All the cells inside were dead. Now he went into the fourth stage. This is really amazing what he did here. He again took the heat-killed smooth cells, like in the previous stage. He took the heat-killed cells. Now all the cells were killed. Now you must always realize that he had conducted all of these stages multiple number of times just to confirm whatever he was doing was accurate, was consistent, right? The outcomes were consistent. So in the fourth stage, what this guy did, he took the heat-killed smooth cells and he collected them in this test tube. So he was ready with heat-killed smooth cells. And he took another Petri plate, a separate Petri plate on which he cultivated living rough cells. Now this plate had living rough cells, the avirulent cells, avirulent. Now the cells that he grew here were rough and they they were non-disease causing streptococcal pneumonia bacteria. And what he did was he poured all of these heat-killed smooth cells, he poured them here into this Petri plate. And in this Petri plate, we already had these living rough cells. Then he allowed this particular heat-killed smooth cell solution or suspension to stay inside the Petri plate for a certain number of hours. And then after a couple of hours, what he did, he scraped the content out. He took the content out of this Petri plate. So now, what would you assume the Petri plate will have? The Petri plate will have these, let us call them the rough streptococcal cells. So I'm now drawing the cell, not the colony. So these are rough streptococcal cells. They do not have a polysaccharide layer around, right? And also, what else does this Petri plate have? It also has the suspension, the dead content, right? There it had the dead content from the heat-killed S cells, right? This is what we'll expect, right? So now he took this suspension out, the scraped out suspension, he made a suspension out of it, a solution out of it, put it into the syringe, injected it into the mouse, and the mouse developed the symptoms of pneumonia and it died. The mouse died. And then he also took a certain tissue sample from the body of this dead mouse and he cultivated it. And he figured out that both the colonies, rough and smooth colonies, were isolated from the tissue. Now here the most important one is he was able to isolate the smooth colonies from the tissue. Now look at this fourth stage. Where do we have living smooth colonies? We do not have living smooth cells anywhere at all. So what caused the appearance or reappearance of these living smooth cells inside the mice, inside this particular mouse? What caused them to reappear? And this is from where he concluded something very important. He concluded that from the heat-killed smooth cells, some content or some molecule or some molecules went inside these rough avirulent bacterial cells. Some of these contents from the smooth cells, the dead smooth cells, they came out and went inside the rough non-disease causing streptococcal pneumonial cells and transformed these rough cells into smooth cells. Somehow these living rough cells that were grown in the Petri plate were transformed. And what were they transformed by? They were transformed by the material present in the dead matter, the heat-killed matter that was added to this Petri plate. And what Griffith concluded from this experiment, he concluded that the cells, the bacterial cell, one bacterial cell can basically transform another bacterial cell by the means of a substance called the transforming principle. So what was transforming principle became the question. Now transforming principle certainly was some kind of a molecule, but what molecule was this? So the significance of this experiment was that contents from one bacteria can enter into another bacteria and change its properties, and this is called the transforming principle. So this is our first experiment that we have seen in the series of 3 experiments that had led to the discovery of DNA as the genetic material. We'll move to the second experiment in our next video.